Hello there, welcome back to GCSE Labs. This video will be on eukaryotic and prokaryotic cells as part of the subtopics of cell biology in the GCSE Science specification. So let's begin. So going on to the key specification points, so these are points you have to make note of. Please make note that plant and animal cells, which are eukaryotic cells, have a cell membrane, a cytoplasm, and the genetic material, which is the DNA, is enclosed in a nucleus. Now, this word enclosed is a really key word. It's a very AQA word because it describes exactly how the genetic material and where it is positioned within the cell. So always use this word in the exam or whenever talking about how genetic material is stored in the nucleus by being enclosed in it as part of the eukaryotic cells. Moving on, bacterial cells, which are prokaryotic, however, are much more miniature or smaller and simpler than these eukaryotic cells. So this point is really significant for when making comparisons between eukaryotic and prokaryotic cells. Furthermore, they all also have cytoplasm as well as a cell membrane surrounded by a cell wall, and we're still talking about bacterial cells. On the other hand, however, the genetic material, which is the DNA, is not enclosed in a nucleus. So we can see that there is a clear opposition between a prokaryotic cell and a eukaryotic cell regarding their storage of genetic material and how it is really stored. And it is a single DNA loop that floats freely in the cytoplasm instead. So it is a circular strand, you can say. A circular strand. There we go. And there would probably be one or more small rings of DNA called plasmids, or you can call them plasmid rings. Now, you might have already identified that this is a diagram of a prokaryotic cell because we got these key words: the nucleoid, which is the DNA, and the plasmid. So we know that the nucleoid, we can see that there is no nucleus. It's like one giant loop within the cell, which is where all the genetic material would be stored. So therefore, we can see that it's floating in the cytoplasm. There, it's not enclosed in anything. And for the plasmid ring, there's only one. And remember that it can be one or more. And this is also another area where the DNA can be stored. You do need to know what the cytoplasm is and the ribosomes, as well as the flagellae, the cell wall, and the cell membrane. Now it says plasma membrane, but it's more preferable to say cell membrane because that's what AQA would expect. You don't need to know what a capsule is or what a pilus is. However, just for extra information, a pilus or pilus are hair-like projections that strengthen the adherence of the cell, which in other words just means how sticky it is towards another surface. So don't forget that this is a bacterial cell that we're looking at, and these are usually accountable for infections, and we can already see that pilus already is a factor for why these infections begin, because they stick to the surface of something. And a capsule just acts as the case for the cell, kind of like a cell wall. Moving on, we have eukaryotic cells, and the main ones we're just going to look at for today are animal cells and plant cells. So the diagrams may look complicated, but it won't seem that complicated once we go over the ones that you don't need to know, and when you just make a note of what you actually have to know. As you can already see, we have the vacuole, the cell, and the chloroplast for the plant cell in green. This is because these three things are only found in the plant cell between it and the animal cell. So these three, so the vacuole, the cell wall, and the chloroplast are not found in the animal cell. And same with the centriole. It is only found in the animal cell and not in the plant cell. And you do need to know what these are, but in our next video we'll go over the animal and the plant cell, so no need to worry about it. They both have the mitochondrion. They both have plasma membrane. Again, it's better to say cell membrane. They both have cytoplasm. They both have ribosomes as well. And they both have a nucleus. Remember, their genetic material is enclosed in the nucleus, so we can already identify that these are eukaryotic cells. However, I'm going to underline the ones that you don't need to know for your exam, but it will be preferable if you actually knew their function. So it's a lysosome, centriole, a Golgi apparatus, and an endoplasmic reticulum. Now this is just where there is a protein synthesis and no wait. Oh yes, it's correct. So there is protein synthesis. So protein synthesis that happens, as well as lipid metabolism. Golgi apparatus, this is a component that stores protein that's ready to be exported outside of the cell. A lysosome is where all the enzymes are stored so that they can break down proteins or polymers. Centriole is what organizes the microtubules that deal with the skeletal system of the cell. You can make notes of the components that need to go around the cells and label the diagrams. However you like to do it would be perfect. Okay, so now have a go at this question. Describe two differences between a prokaryotic cell and a eukaryotic cell, which is 2 marks. Now I'm not too sure why I put a question mark, so just ignore that. Because this is a command word, not a question word. Yes, worth 2 marks, and I'll let you annotate this question first, and then we can go and see what a model answer would look like. Okay, so now we can see that it says describe and it's in orange. This is because describe is the command word, and we always have to look out for what the command word is actually is. And describe always refers to what. We always state what something is like or what it is. So we're not given any reasons for anything. We're just talking about the what. The word two is really key, because if someone only does one difference, then we're likely just to get 1 mark, because we have to correspond what the question is asking to to how many marks there actually are available. So 2 differences, 2 marks, so we can already identify that 1 difference will mean 1 mark. Now I put in orange prokaryotic and eukaryotic so that we know what we are actually comparing. So have a go at this question, spend no more than 3 minutes, and we'll go over what an actual model answer is. Okay, so now let's read the question again and go over the model answer. Describe 2 differences between a prokaryotic cell and a eukaryotic cell. 2 marks. Now this is what a model answer would look like. If you have anything similar to this, or maybe possibly the same, then you are on your way to getting both those marks and above for any other related question. One difference is that a prokaryotic cell, for example a bacterial cell, consists of the genetic material being in the form of a single circular strand that floats freely within the cytoplasm and is not enclosed in a nucleus like a eukaryotic cell is. Also, prokaryotic cells have a much smaller and simpler structure than eukaryotic cells. And as you can see, in brackets I inserted where my one markers would be gained. And if you noticed, after every difference that I mentioned, there will be 1 mark, and the second difference, 1 mark. This is also acceptable. There is no mitochondria in prokaryotic cells, yet there is in eukaryotic cells. That also would have gotten me 1 mark. Now, it's really important to actually match how you answer the question with what the specification actually provides for you to obtain as part of the curriculum. Now, I hope you took what you need to from this video. Stay updated for the next video on animal and plant cells, and I'll see you next time. Bye bye!